Hello, everyone, and thank you for joining today's Indica Labs webinar. My name is Ben Dyer, and I'll be the moderator for today's event. If this is your first Indica Labs webinar, I'd like to let you know that you've come into the webinar on mute and will remain on mute for the duration of the session. If you have a question at any point during the webinar today, please write it down in the questions panel of your webinar controls, and at the end of the webinar today, we'll have a Q&A session with our presenter. Now it's my pleasure to introduce Dr. Ghislaine Liu, our presenter for today's webinar on multiplex fluorescence image analysis in HALO. Ghislaine joined Indica Labs in 2020 after earning her PhD in molecular biology from the Autonomous University of Madrid, and is currently a senior field application scientist with us. As part of her role, Ghislaine works with customers to optimize their image analysis workflows and find the analysis solutions that best fit their needs. During today's webinar, Ghislaine will discuss and demonstrate key digital pathology workflows for fluorescence images, including tips and tricks to help improve your analysis. Ghislaine, thanks for sharing with us today. I'm looking forward to a refresher on the fundamentals of multiplex fluorescence image analysis in HALO, and now I'm happy to turn the presentation over to you.
Thanks a lot, Ben, for your kind introduction, and welcome everyone to this webinar about getting started with multiplex fluorescence image analysis in HALO. So I meant to say, first off, that this webinar is not a masterclass, but rather an introduction to multiplexing workflows. So this is aimed at those who are new to these technologies or considering adopting them. So let's start with today's agenda. I will first introduce our software ecosystem, and we'll then cover the fundamentals of fluorescence multiplexing imaging and explore the key technologies available. I'll also guide you through how to intuitively analyze multiplex fluorescence data using the HALO software. And this session will include a live demonstration and a Q&A at the end. As you know, HALO is our image analysis powerhouse, and it is scalable. So it means that various analysis modules can be chosen depending on the applications of your groups, such as cell segmentation algorithm, tissue classification, serial section alignment, and so on. Additionally, we have HALO AI, which is our artificial intelligence add-on that allow users to train the software to recognize any structure the human eye can recognize, such as glomeruli in the kidney, segmenting the tumor tissue from the stroma, and even doing it at the cell level. So our users can save time. Our teams also have created AI apps, which are a suite of pre-trained algorithms tailored for specific needs. For example, identifying tumor cells or tumor tissue in different cancer types, such as lung carcinoma, breast, or gastric cancer. And we then have two image management software. So HALO Link for research and HALO EP for clinical pathology workflows. So while HALO is installed on a workstation usually, HALO Link and HALO EP are browser-based. HALO Link connects directly with HALO and facilitates collaboration through annotating or running saved analysis according to specific permission levels, while HALO AP performs case-centric slide management, so users can transition validated assays from HALO and run them in a locked-down CIVD environment. And last but not least, we have a pharma services team of image analysis experts that can leverage all these platforms to perform image analysis for customers who have more data than they can analyze in a given time. So let's now discuss the importance of multiplexing technologies, taking the example of the tumor microenvironment in the field of immuno-oncology. So as you may know, the tumor microenvironment is the seat of multiple cell interactions between tumoral cells, stromal cells, and immune cells. And those interactions have been shown not to only affect tumor aggressiveness, but also patient prognosis. So being able to evaluate 40 markers or plus at the time on a 2D image is a great advantage for understanding tumor identity and be an important step towards therapy design. And as the field is trending in the direction of added markers, there have been a few technologies to support this. So I will briefly mention a few hyperplexing techniques. These technologies range from the use of bright field, fluorescence, antibody staining and stripping technologies, as well as metal-tagged antibodies in the field of image mass cytometry. And we are proud these technologies are all supported for analysis by HALO, as these have significantly influenced development at Indica Labs. So let's now discuss what these datasets involved from an image analysis perspective. I'll now dive into all the options that are available in Halo, and I will take again the example of the tumor microenvironment. So here are a few steps one may follow when exploring this type of images. The first thing you may want to do is to categorize the tissue into compartments automatically and reproducibly, such as the tumor here in red and the stroma here in the middle. And for this, you can use our tissue classification algorithm as an example here. And this will then allow you in a second step to focus on the biomarker evaluation. So the idea here is to phenotype cells into your negative, single positive, double positive, and triple positive cells. So while being actually able to tell what compartment every cell belongs to. And once all of the cells objects have been identified, you also have the option to explore the distribution of the cells via our spatial analysis tools, such as the proximity analysis tool, the nearest neighbor analysis, density heat map, and so on. And finally, I'd like to mention the last step you may want to take, which is to identify groups of cells in an unsupervised fashion with our brand new clustering module. During the demo phase of our webinar today, I will mention all the different steps here, but I will mostly focus on the biomarker evaluation phase and touch upon the clustering workflow, as the tissue classifier and the spatial analysis workflows have been described extensively in our past webinars. So moving on into the Halo ecosystem, let's check the different tools I will be using during the live demo today. And at the center of the analysis, we have our multiplex imaging analysis module here, which is Hyplex FL. So we'll be using the Hyplex FL analysis module to segment cells and phenotype them, as you can see in the real-time tuning window here. And Hyplex FL, as other cell segmentation algorithms now in Halo, can leverage our nuclear and membrane pre-trained networks for accurate and automated detection of cells, while in the past, only Halo AI users could use artificial intelligence for this. So we are quite proud to have this included now. And prior to exploring your biomarkers, you may want to assess your image cohort heterogeneity in terms of intensity. So you can now do so using our new view settings that allow you to check the intensity graphs of several images at a time. You will then want to find your positive cells. Since our latest 4.1 version, Halo users can use the threshold assist tool to set positive thresholds automatically. In the past, you had to set those thresholds manually, but instead, this can now be used to threshold an unlimited number of markers automatically, basically. And once you have identified your positive cells, you will then want to leverage our phenotype capabilities. So we do have a new phenotype editor tool that allows you to create phenotypes easily by providing a name and adding basically a plus or a minus logic to the channels of interest. And finally, to explore your cell data in an unsupervised fashion, identify cell groups you may or actually may not have thought of, you can use our brand new clustering module. So there are obviously more options. I'm not going to describe them today, so I don't want to overload this presentation, but just so you know, you can export your object data as FCS and CSV, and you can actually process them in other software like FlowJo and re-import them if needed into the software for a bilateral connection between the object data and your actual biological image. So I'm going to dedicate a slide to briefly explain what clustering and dimensionality reductions are, since they may be new concepts for people starting with the analysis of multiplex images. Clustering is something we actually all do regularly at home or not, and when we are sorting out items into functional groups. So for example, here, the functional items would be my t-shirts on one side, my shoes, my books, and for example, kitchen items. And high dimensionality reduction on the other side does not actually change the grouping itself, but it allows you to visualize all objects in a way that is easily readable for the human eye. So this works exactly the same way as your furniture at home. And so how does this work in practice? Here we have a UMAP, which is our dimensionality reduction plot that allows us to visualize all items. So these are the clothes items I mentioned earlier. And each color here represents a group, so an identified cluster, sorry. So obviously in Halo, your objects won't be shoes anymore, but cell objects instead. But the principle is exactly the same. You will be looking at different cell features, so the properties that identify your cells, basically, such as cell intensity, nuclear shape, location, and so on, so that Halo tidies up those objects into readable plots and reveals cell groups that can then be identified directly onto the biological image as well. So every single dot here basically is a single cluster, well, every single color is a cluster here. That said, I would like to finish off my PowerPoint presentation with a little bonus for our Halo AI users that I haven't mentioned yet. Okay, sometimes when it's not possible to phenotype cells because of the quality of your staining or because your cells of interest express a marker transiently, you can always train our artificial intelligence add-on to recognize the cells of interest on the sole basis of their nuclear pattern, for example. So I've purposely taken the example of an IMC image from standard biotools to illustrate that the Halo multiplex workflow is also valid for IMC images as well. And here you can see that the software was trained to identify and distinguish tumor cells here in blue from the stromal cells here in magenta on the sole basis of the DNA pattern. So it's not even like, it's not even antibody at that point, it's just DNA. So that pattern was basically enough to find out on a pattern-based fashion where those cells are. And I hope you can see how this AI tool can definitely be of added value. And I'll take a couple of minutes to showcase this at the end of my demo today. Okay, so let's now hop off to the demo of the software. So I'm just going to move on to Halo. And we're going to move on to these data sets that I have here in Halo. So this data set is colorectal cancer from Luna 4, actually. And the reason why I've chosen those images is because they reflect quite well the problematic, the issues that you can get or the challenges you'll have when working with 20-plex plus images. So this is actually a 20-plex image. And the first thing that that one can see is that this image basically looks pretty white. And the reason for this is because we have so many channels that when they overlap with each other, basically the resulting color is white. And with this, you can't really see much, can you? Well, actually in Halo, so I'm cheating a little bit. I have already done that work before. So I'm just going to tile those tabs vertically. So this is an image I processed by changing the view settings in Halo from the same data set, and this is an unprocessed image. But the idea here is the same. So you go to the view settings here in Halo, and you basically change the brightness and the contrast. So here I'm changing the contrast with changing the blacking value, and I can do this on a per channel basis. So that you don't see me doing this for like 10 minutes. I'm just going to do this. I've done this already once here. And you can basically apply all these settings in batch throughout the whole data set. The only thing you have to do is go to the view settings again and go to these settings actions, batch apply settings, and tell Halo what settings should be applied and on what image. I have an option here that is quite interesting, is to apply channel names. The reason this is interesting here is that you may have noticed that, actually, I'm just going to cancel for a second. You may have noticed that those channel names are pretty crowded. So I wanted something a bit more efficient for my eye and relevant. So I actually changed the channel names by just right-clicking here and changing those names here. And I can also carry this through the view settings. So I just apply my batch, my settings in batch, apply my channel names as well on this image here by hitting apply, and you see in a couple of seconds that this has been applied properly. So I'm just going to, so here you see that this image has now been, the view settings have been changed. It doesn't actually affect any metadata of this image and those channel names as well. So other things we can do as well is to go to different images and go to the view settings again. And you'll see that if I go to open comparison chart, I can actually select various images so that I can visualize how the histograms of my intensities look onto these different images. So this is a great way. So if I just scroll in, you see I can compare how this looks in different images, and I can just check that this doesn't look off in one of the image. So if I have, for example, one outlier, I may want to create new settings for that outlier. So this should be pretty homogeneous, so I can go ahead and move on to the analysis. So I'm just going to focus on that image and close all tabs but this one. All right, so here we have a few things. We have the tumor cells here depicted with the PAM-CK marker in red, and we have the stroma and some immune cells here. So the obvious question here is how many cells I have for each compartment, what is the area of each compartment, and am I allowed to analyze everything without processing my image at all. So actually, when I moved on to one of my images before, there is one thing I saw, which is that I have some out of focus areas. I have also some red blood cells, and this I need to be very careful about this because these may actually account for a false positive signal into my analysis when the time comes. So there are actually ways to get rid of this before I start my analysis, which is to use a classifier. So I mentioned that we do have pattern-based recognition in Halo. We have classifiers. I have trained a classifier already, and I'm going to show you our real-time tuning window, which is our very powerful tool in Halo that allows you to see in real time what the software sees, basically. So based on this classifier that was already trained, you can see that the artifacts are actually recognized in white here. I can actually change how this is being depicted. So you can see that I can change transparency. And if I go around, you can see that all my red blood cells are being removed here in the white area. And everything that is left is the red area. So this is going to be a really good idea for me to pre-process my tissue with this classifier before I start. Another thing I want to do maybe is to quantify how much tumor and stroma I have in terms of area and percentages. So for this, I can also use a classifier. So I already have a classifier saved here. Here it is. And if I put back the real-time tuning window again, you can see that the tumor is being depicted in yellow and the stroma is being depicted in blue. So it's actually quite straightforward to see that this was trained properly, and this is going to be actually quite relevant for my analysis then when I have to move on to the cell segmentation. So since Halo 4.0, we can actually put those classifiers into a pipeline. So this is something I can do so that I don't have to multiply the steps, the pre-processing step. So I could have, like, for example, create a classifier pipeline, add, for example, my first step as the artifact classifier, just remove anything that is not tissue here, then add another step, take my tumor classifier, select everything that is not backgrounds, such as tumor and stroma here, or tumor and stroma, and then have this saved as a classifier pipeline. I actually have already saved this as artifact removal and tumor stroma, so that I can then use it at the stage of cell segmentation. All right, so I don't have to worry about this anymore, and I'm going to move on to the biomarker evaluation. If you ever remember, if you ever wonder how this was trained, we have a substantial amount, of classifier trainings on the learning portal and webinar. So this has been covered quite substantially. Okay, so let's go back to now the biomarker evaluation. So I'm going to use the Hyplex FL module here. And as you can see, the Hyplex FL module actually is already ready to use. So I have the different steps that I have to follow here, which are the analysis magnification, setting up the dyes. So for example, I can autofill my dyes automatically just so they are imported directly from the metadata of my image. And if I want to, I can even sort of remove some that I'm not interested in. So for example, the autofluorescence here is not really relevant for me. So I can remove that specific dye and go back, for example, to the dye selection if I want to remove specific dyes. All right, so now that I have all my dyes inputted, one thing I may want to do is to segment my cells. So the first thing you have to do is to start finding the cells, or sort of the nuclei. Then you grow a cytoplasm around those nuclei. And if you have a membrane dye, then you can use that membrane dye as well. So I'm just going to go back to this image here. because we have a nice portion of stroma and tumor. And here, first off, what the software does is to segment cells. But this segmentation may not be so robust because I may have to change, for example, the contrast threshold parameters, the intensity parameters to filter the aggressiveness of my segmentation. And I may have to spend a few minutes to do that. Or I can also use our AI workflow. And in a couple of seconds, I can have a segmentation that looks much better. So as you can see here, if I put back the DAPI and change the color of the DAPI and make this more transparent, just so you don't have to trust me, basically, we can go around and you see that the segmentation looks much better. And I've spent like 1 minute on this. So obviously I still have access to different filters such as nuclear size, segmentation aggressiveness, and so on, but I pretty much don't need to do anything by now. Okay, in the next phase, so I'm just going to put this back into blue and put back all my channels. Into the next phase, what you may want to do is to set up a cytoplasm radius. So let's go to maybe some proteins that have, where we can actually see the cell shape. So I can go ahead and go to my next section and say that I want to set up a cytoplasm radius. So let's say 1.5, for example. So I did write 1.5, but for some reason, it takes a bit of time, and it always does when you're presenting a webinar, obviously. So please forgive me for that and give me a couple of seconds just so this comes back. All right. If it doesn't, yeah, oh, perfect. I have to keep faith. Okay, so now we have the cytoplasm radius that has been drawn here. And basically it has the radius I've just inputted here. Where it becomes problematic is when I have big cells and I have cells which have a like huge cytoplasm, right? So in that specific case, because I have a membrane dye, so I have the CK membrane dye, I could as well just use our AI mode for the segmentation of our membranes based on the membrane dye here. So just give it a couple of seconds until it updates real-time tuning. But basically, you must see in a few seconds that the membrane segmentation will appear. And if it doesn't, then I will just reload some settings. All right, so I may have to load, just give me a second, some settings I already have here, just so you see how this looks like in practice. So here, membrane, segmentation type, AI. Okay, here it is. It was too shy. Okay, here. So here you see that instead of having like a round shape here, we do actually have the membrane being depicted here. All right, so we have actually gone forward a bit too much in the sense that those have already been thresholded upon, but I can show you how to do this right now. So these, in the past, you had to threshold them by adding a value. So you had to set up manually the thresholds. But now we do not actually have to do this. We can actually go to the Studies tab, find out some results that I have already run. So if I go back to my results and find my unthresholded results, I can actually use unthresholded results and use our tool that is called Threshold Assist. Okay, so our threshold assist tool, what it will do will basically to be finding the threshold. So I'm just going to show you the real-time tuning at the same time, just so you see what it does. So actually, okay. So as I move on to the different dyes, you basically see that it finds my positive cells automatically based on the intensity information of all single cells of all my images in this experiment. So it's a really data exploration approach, and it allows me to segment those cells and threshold them upon in an unbiased fashion. And if you want to verify that this is actually correct, you can always go back to, for example, the channel of interest. So maybe so that it's easier to see, I'm just going to go to an absorption mode. So this is a mode you can use to look at cells in a pseudo bright field fashion. And you basically see that all those cells are being recognized quite correctly here. You can also choose what compartment is being thresholded upon. So if I go, for example, to our CK, just so it's more striking, you see that now here, so if I go to some CK signal, you see that we do see the signal in the nucleus, which doesn't make sense, right? So you can actually change this and go to my membrane instead, and you see that the membrane is being highlighted now in yellow. So this is basically just how it works. So in a few minutes, you just go to the different channels, pick up the compartment, and that's it. So this is something I've already done. So I'm just going to reload the settings again, because obviously every time I use threshold assist, it goes back to the unthresholded data. And you're going to see that this is actually, so if I just go back here and put all the channels back and put the real-time tuning window, this is actually the image with all channels together. So this, it's very colorful, and this is because all the channels are blended together. But do not worry if you have many channels. You may want to use the interactive markups, which we have in Halo. So it will allow you to explore the single positivity at the time on the markup. All right, so now that we have our cell positivity, so you see all of these have been thresholded upon, so it's all good. I can move on to this most exciting part, which is to set up phenotypes. So if I go to my phenotypes here, you can actually see that I have set some up. And once I want to add a new one, I just have to go to add new phenotype. Or I could duplicate the last phenotype. So here we have macrophages here. So let's create a new phenotype, which is my M2-like, because the first one was M1-like. And as you can see, it's going to be pretty straightforward. You just need to choose the logic and/or, and basically set up the positivity or negativity for the markers of interest. So CD68 would be fine for M2-like. Let's make it negative for CD163 instead, and positive for HLA-DR. And once I'm done, I can just press OK, and that's it. All right, so now that I have my phenotypes, I can go ahead with my analysis. But just before I move on to the analysis, I'd like to mention a couple of things. So you remember we've set up an artifact removal. I had a classifier for artifact removal and for tumor recognition. You can actually include that classifier pipeline in these features or in these advanced parameters, just so this can be used to pre-process the image before the cell segmentation is being run. So then I'm sure that my artifacts are being removed and the cell segmentation is taking place only in a tumor and stroma. Another thing I can do since 4.0 is to actually customize my outputs. So these are all the outputs you get. So the total number of cells, positive cells, you have the phenotypes as well, the compartments that are involved in the positivity. You have here the percentages of positive cells and their intensities as well. And because I'm using a classifier, I have also all the information just for the stroma and all the information as well for the tumor that is being separated. And one thing I can do as well is to create calculations so that I can output those without having to go to Excel. So let's, for example, go to the density calculation. Let's say that I'm interested in my CK, well, let's say here, sorry about this, my CD31 positive cells. So this is a very random, but it's just an example. Okay, so this out of my area analyzed. So here I can just basically create a custom calculation out of the box just that way. And I can just save those settings and apply them. Obviously, I've already applied those settings, so we don't lose time here. But basically, this is how this looks on the real-world image. So you see that we do have all cells segmented, all the colors appear here. All my output data appear in the results up here as summary results, and all the object data down here. And the nice thing about object data is that you can actually go ahead and move to different cells here and actually look exactly what are their properties. So you see their intensities, their metrics, like cell metrics, like size, area, the type of label. So for example, here, this one is localized in my tumor, while this one maybe is localized in my stroma. And you can look at the ID and the coordinates. So for people who want to just export this out of Halo right away. This can be done at any point. So you can just export everything here as a CSV or FCS, or you can also just process everything in Halo. So let's imagine that now I wanted to filter my cells and look where they are in my image. So let's filter my tumor cells, and let's select them all into my object table. So here my tumor cells appear on my real world image, and I can use this to QC my data. If I do have the spatial analysis tool, one thing I can do as well is to just go at a quick glance, go to plot, spatial plot, give a name to my cells of interest, and have them plotted in a few seconds as well. So you can put an unlimited number of markers. This is just one as an example, but this can be done at any point. Okay, so as I mentioned, this is maybe a bit crowded. If you have all the colors together, you may not know what's what on this mask. So for publications, what you may want to do instead is to export an interactive markup instead. And this is where it becomes really pretty colorful because every color here is color attributed to our interactive markup. So I've actually taken out our markup legend from here, and you can see that all those channels, or all these colors actually, can be toggled on and off at any point. So this is actually pretty handy, especially if you have many channels, don't want to see everything at the same time, and you want to QC your data. So this is the way you want to do this. You can do this for all cells in general, but you can also do this for phenotypes as well. So this is just one interactive markup for phenotypes. And in those phenotypes, you do see that it does look a bit different because instead of having the single channels, we now have the phenotypes. So if I were to toggle this off, I can now move on to my different phenotypes and have a look at them all together. So when it becomes interesting is for publication, right? So we have actually a figure maker we've already had before, but this figure maker has been renewed so that everything is a bit more smooth, and you can save those figures internally into Halo until you are ready. So if I were to use this figure maker now, I could, for example, toggle this off, zoom in my image, select this field of view. Then, for example, say that I have one row and 3 columns, and then, for example, just phenotype my T-Rex, just show my T-Rex or any, sorry, T-Rex here on a cell type that is a bit more visual. I can more visualize here. So my tumor cells, for example, here, and let's go back, for example, to another result so I can have as many results as I want really in this. And for example, take my colors for the markup here for all the cells of interest or all the dyes of interest. We can even zoom in here and have, for example, my scale bars, reference frame as well, and let's say alpha frame titles as well. Okay, so if I'm done now, I can just save this. And whenever I'm ready, I can download it basically. So if I want to open an old figure, it's here, down here. All right, so now we are basically pretty much done with the cell segmentation workflow, but there are a few things that you may want to do, right? So you can obviously just have all these analyses that have been saved and run them in batch. So if I just go to the studies tab again, where everything is batchable, I can select my images of interest doing Control A, image actions and analyze, and have everything analyzed with the same settings. And once everything is done, I can also go to my export functionality and try to export any results that make sense or that I need to have. So here again, all my images that need to be exported, CSV file, FCS file, any object data, anything needed. But there are other steps that you can do in terms of data exploration. So we have sort of opened a new field of data exploration in Halo, while before we were just focusing on image processing, which is our clustering and dimensionality reduction workflow. So this is accessible at any point now in Halo, as soon as you have some object data from cell segmentation module. So if I go to the image actions here, and I just go back to my results like before, and I just filter them back and select them here, I can go to cluster, and here I'll have this new window popping in where I can actually decide what features need to be taken into account for my clustering and what algorithm should be used. I can also define a number of clusters for k-means, or I can have the program decide of the number of clusters on its own. So this is something I've already run before, just so we don't run out of time. And I do have a k-means here. So I'm just going to move on maybe to this image instead. And right away, when the k-means graph appears, what you see is you have those dots that appear directly onto the image. So this UMap here shows you, like the UMap here is a 2D plot, right? The clusters, however, are the colors on your cluster, on your plot. So you have an interactivity between those cells here and the cells here on your image. If at one glance you want to have an idea about the cells that you have and the groups you have, you see that we do have more or less our tumor cells here in yellow, our immune cells here in red, and our stromal cells in blue, potentially, right? But if you wanted to check this, you have different ways to do this. So you can either look at the intensity information. So if I go to the CK intensity for cluster 1, it looks again like there is a peak for CK, so it looks like tumor. But where it becomes interesting is where I can actually go to my graph here, or my UMap, and have this little pen draw a region that I can then have reflected directly on top of my image. So these are a lot of object data. So maybe that's why it's taking a bit longer. But technically, so maybe if I move on to a different one, just so it reacts. Or maybe just let's close this again and just reopen. Sorry about this. It's going to take a couple of seconds to do this again. Here it is. Here it is. Let's zoom in. Let's draw. And here we are. So this is what I wanted to show you. So basically, when you draw onto the UMap, you can actually have those cells identified directly onto your biological image. And as you can see here, it does seem to actually match on the thumbnail here, my tumor cells here as well. You can obviously change those names as well of the cluster, and you can call them tumor cells once you know that they are tumor cells indeed. All right, so now that I've shown the k-means, I just wanted to check really quickly the phenograph, just so you see that there are different methodologies and they look a little bit different. So here you see that we have many more colors, because what phenograph does is actually to find the clusters itself. So before I decided on 3 clusters because I was expecting to have tumor, stroma, and immune cells, but in fact you do have subpopulation that may be interesting for data exploration for your experiment here. And actually, even just in the tumor environment, you see, or in the tumor itself, it seems that there are subpopulations that may be of interest here. They may not be reacting all the same way. All right, so okay, so last but not least, what I would like to show you is the phenotyping workflow as a bonus, really, because as of now, basically with Halo and the multiplex algorithms, everything that I've done can be done. But if you have Halo AI, there are certain things that you can do as well. So I'm just going to move on to here some image that I'm going to use for with a classifier here, and I'm just going to bring a phenotyper here in the classifier tab. So sorry about this. Classifier actions, load. Here it is. All right, so I've already preset the tumor and over class, basically, because the idea here is to train the classifier or the object phenotyper to find my tumor cells on the sole basis of the DA or the DAPI, sorry. So you can actually check this by going to the advanced options. And as you can see, I'm telling the software to look only at DAPI. So you need to keep an open mind, obviously, here, because this is for demo purposes. It's not going to be perfect, but it's just to show you how powerful the software is really at recognizing cells just based on one color, just based on pattern of the nucleus, for example, in that specific case. So what I've done here is to start the segmentation just so you would see what Halo uses as a segmentation pre-trained algorithm. And as soon as I have started the segmentation, I can take this highlighter tool, select the class of interest, and select my cells directly onto the image. So it's just as simple as this. So I'm going to do something a bit like, it's not going to be super accurate, but it's just for demo purposes. But it's just so you see the principle of what I'm doing here. So basically, every time I shade over the cells of interest, I basically have annotations being fed to the network here. So every single row is a single annotation, a single cell. And the idea is to have cells for each compartment. So let's, for example, have some example of immune, some example of tumor, sorry, some example here again of tumor, and let's put the other bit anywhere here. Let's just make sense. Just make sure that it makes sense. Here, here, and here. Okay, so for demo purposes, this will be enough. And the next step is going to be to start the training. So I'm just going to hit start training, and I'm going to put up the real-time tuning window, which now is actually on the entire field of view. And what you do see here is the number of iteration, which is going up. So the number of iterations is the number of times. The network sees our annotations. And the cross entropy, which is the value that is going down here, is the difference between our annotation and what the software classifies. So we do actually that this value is going down. So obviously this is going from interpretation to interpretation, but we do have some trend already. So you do see that we have obviously some mistakes still, but there are some regions where the classifier has already figured out that these, for example, are tumor cells. Here, these are mostly tumor cells as well. These are stromal cells and so on. So the idea to make it perfect is to let it train for longer, but I think it's already quite impressive, like in just a minute, that is capable of finding out most of my tumor cells just out of the DAPI stain. Okay, so I'll be done here for now. I'll move on now to the Q&A. And before we move on to the Q&A, I'm just going to go here. And I'm going to let Ben take back control to talk about the upcoming webinars and talks.
All right, thank you so much, Ghislaine, for a great presentation and demo today. Before we move into question and answer, like Ghislaine mentioned, I'd like to briefly touch on upcoming events. If you do have a question for Ghislaine, now would be a great time to write those down in the questions panel. We already have a few, but keep them coming. For upcoming conferences, we're looking forward to the European Congress on Digital Pathology in Barcelona on June 25th to 28th. The team from Indica Labs will be in booth 19, and we have 2 oral presentations at this event presented by Dr. Peter Kai, Senior Principal Scientist who leads our AI DX team, AI Diagnostics team, that is. Peter will be presenting on how breast IC AI can be used as a training tool for students and will present a multi-site analytical performance validation of our Slide QC BF product. Additionally, Peter will be sharing about how our clinical AI software can add efficiency and quality to your digital research and clinical pathology laboratory during the AI innovation pitch session on the 27th. For webinars, today's session is the last before our summer holiday. We wish everyone a relaxing holiday and safe travel during the season and look forward to seeing you again after the break. So now moving into our Q&A session. Like I mentioned, we already have a few questions. Go ahead and send in any more you have. But to start off with, we'll do a bit of housekeeping here from inside the building, so to speak. And Ghislaine, first question for you is there a free trial version or how can someone test Halo and Halo AI out somewhere?
Yeah, of course. So we do actually offer free trials for 30 days, and it means that you have exactly the same version of Halo as our customers and the same service as well. So our customers have unlimited support if you have annual licenses or are covered by maintenance. And basically, it means that for 30 days, we'll be installing all the modules that you need to review, AI included, and we'll be also training you and helping you throughout.
Fantastic. Thanks for running through that option, Ghislaine. And anyone who's interested, please do feel free to reach out to us at info@indicalab.com or to your local application scientist if you have that connection already. Now, the next question, Ghislaine, coming in is a question about the phenotype setup. Um, And a question about the logic in phenotypes, whether a cell can belong to two different phenotypes and how the and or logic kind of works when you're setting up phenotypes.
Yeah, that's correct. Actually, two cells can be independently linked into two different phenotypes because every single phenotype is independent from another. And it means that when, for example, you have N or, means that, for example, if you have A or B in your phenotype, then all cells A and all cells B basically will be present in this phenotype. If you have A and B, it means that they need to be double positive to be counted into the phenotype. I hope that this addresses the question.
Great, thanks, Ghislaine. And if not, ask her, please feel free to reach out again in the Q&A if we didn't quite hit on that. Next question, Ghislaine, is asking about the blood vessel and autofluorescence detection step. Asking for that classifier, were you training on all channels or only on specific channels?
This is really up to you, actually. In this specific case, I did train on the autofluorescence channels, and then all the channels that I thought were sort of more highly autofluorescent, let's say, as compared to the background, really. But you could have as many channels as you want. This really, like the channels you choose need to be relevant for making the difference between one class and another. So you want to have a difference of intensity between the different classes you're trying to categorize.
Got it. Thanks for running through those considerations, Ghislaine. And let's see, next question is about um, pixel values here and asking, how do you find the fluorescence intensity data associated with each object or cell? And ask her if we didn't quite get your question here, please, again, let us know, give us clarifying information there. But asking about, yeah, basically trying to find the specific information on objects or cells in your analysis, Ghislaine, and how you'd find that fluorescence
Mhm.
intensity data.
Yeah. So basically, there are two things, the input and the output, right? The input is going to be what the threshold assist sees. So our tool is going to take the surface or the whole area of the cell, and is going to look for the average intensity throughout the whole cell. This is pulled out together into a plot. So if when you have like all these intensity values from different images, these are all computed together just so the threshold assist finds the correct intensity or the recommended intensity that you need to use as a threshold. As an output on the other side, you have the possibility to output those intensity values as well in an untouched fashion. So for example, you'll have the average intensity outputted on a per cell basis, as well as on a per compartment basis. So for example, in the nucleus only, in the cytoplasm only, and in the membrane only. So this gives you a lot of freedom at the moment of processing your data in case you don't, or well, if the gating was not enough or if the thresholding it was not enough, you can always post-process your data and check those naive intensities as well outside of Halo.
All right, thanks for the detailed answer there, Ghislaine. And the next question is asking if the only way to identify an artifact is by fluorescence signal in more than one channel, can AI use this as a basis for exclusion?
Yes, so actually, the classifier I trained was an AI classifier to recognize those artifacts. So I did train those classifiers in the past on several images. I even had one classifier working out of like TMAs, whole slide images, and like maybe I don't know, maybe 10 or 100 slides. It was working on all of them. So as long as you have enough training, basically you can use AI for artifact removal. And in terms of other tools than AI. We do have other tools, but for our bright field module. So we do have like ways to pick up artifacts based on the stain or the color.
Gotcha. Okay. Well, thanks for, yeah, that guidance there and info, Ghislaine. Let's see, the next question here, we're getting a lot, so thanks, folks. I have plenty of time to answer these, so keep them coming. Question here is about a attendee who's already running and doing some analysis here in Halo, and question about maybe some optimization. So the question is, I'm looking at some expression levels of candidate biomarkers of breast cancer, looking at RNA-scope 4-plex assay fluorescent images. For my analysis, I've used the FISH module, which seems to be working well. Would using the current module, which you talked about in the webinar, would that be helpful? Would it be more beneficial?
Actually not, in the sense that you're looking at RNA-scope signal, and the module I've used, the high-plex FL module, is only looking at protein signal. Although most of the functionalities that are present in the high-plex FL module that I've mentioned today, so for example, the AI capabilities for nuclear segmentation, membrane segmentation, are also present in the FISH module. And you can also leverage other AI algorithms with the FISH module, as well as you would, the same way as you would do in high-plex FL. So if you need help for that, me or my team would be really happy to connect in another session and check this with you.
All right, thanks for running through those capabilities, Ghislaine, and the specifics of each module. Um, the next question is it possible to train a classifier that annotates the different brain regions like cortex, hippocampus, etc., automatically?
Um, so I'm going to ask another question. So basically, what you want to make sure of is that you can differentiate yourself those regions automatically with your eyes, right? On a non-local basis. So if you are able to find out, for example, if we give you a field of view of a cortex tissue and you don't have any local information, if you're able to see that this is cortex and this is not, I don't know, hippocampus, then definitely the software can do it. If However, the only sort of information you have to recognize the different regions is on a location basis. So one tissue is next to each other, is on the right, is on the left, and so on. This is not going to work out with the AI algorithm itself because it really needs a different pattern. That said, we have some things in the roadmap, and we are trying to make that work for the future.
All right, Ghislaine, thanks for running through those considerations and yeah, guidance on when AI can work and when it's going to have troubles. Next question for you, Ghislaine, is question about phenotype analysis. And question is, can you only classify cells as positive or negative, or can you also see/visualize high-low populations?
Oh, yeah, that's a really good point. Actually, this is not something I showed today. We do have a masterclass about high-plex FL that actually shows this feature. But you can actually have your data categorized into low, moderate, and strong signal. So you have 3 thresholds. The one I've commented today is just a threshold for positivity. But in fact, after the threshold of positivity, you can have another threshold for moderate positive cells and strong positive cells. So you basically have to set 3 thresholds so the software can compute a H-score for you automatically.
All right, thanks for that rundown and walkthrough there, Ghislaine. Next question for you is capability question about can a Halo AI phenotyper be used on bright field images like H&Es?
Yes, definitely. So the phenotyper, so any AI classifier really isn't modality specific. So you could be using those classifiers onto any type of image modality, fluorescence or bright field, doesn't matter.
Got it. Sounds great, Ghislaine. And then another question is, let's see, is there a maximum number of channels that can be analyzed in Halo? And I'm assuming they're asking about Hyplex FL here, but if you can speak to that.
Mm-hmm. Yes, actually, there is no maximum number of channels that you can analyze into the high-plex FL module in Fluorescence. This can be unlimited in theory. The only downside, obviously, is the more channels you have, the more time Halo will take to process the image, and the more time it will take to analyze your image as well. So this is the only thing you need to take into account.
Got it. Thanks, Ghislaine. And with that, it looks like we've answered most questions that are coming through. If we missed your question and didn't get to it online, we do have those all recorded on our side, and we will follow up by email with anyone that we missed. So thanks everyone for submitting the questions here today. Before we close out, I'd like to thank Ghislaine again for the excellent presentation and demonstration today. And we also appreciate everyone in our audience for joining us. And we look forward to seeing you at one of our upcoming events. So thanks everyone again, and I hope you have a great day. Goodbye.
Thanks, everyone.